Hey everybody, in this video I am going to talk a little bit about the discovery of RNA interference, so RNAi. And in the next video I'll talk more about the detailed mechanism of RNA interference. So before I start talking about how this was discovered, I just want to touch on this term gene silencing, which can be kind of confusing. So gene silencing, what do we mean by gene silencing? So here's just a simple diagram showing how a gene is transcribed into a messenger RNA. Well, at least a protein coding gene is transcribed into a messenger RNA, and that messenger RNA is translated into a protein. So now, gene silencing would refer to like any technique that we can use to reduce the gene product. In this case, the gene product is the protein, right? So any technique we can use to reduce the gene product without mutating the gene. And we can reduce the levels of the product down to nothing, or we can just, reduce them lower than the wild type, the normal levels, and that will count as gene silencing. Okay, so that's just a short definition or explanation on what gene silencing is. And RNA interference is often used to silence genes, so to decrease the amount of gene product without mutating the gene. So the discovery of RNA interference goes back to the early 1990s. And two of the most often cited examples when reading about the history of RNA interference involve studies of petunia and studies of a fungus called Neurospora crassa. So most of you probably can look at that petunia and say, "Oh, that's a flower." Okay, yeah, sure. Maybe petunia looks something like that, but this fungus, Neurospora crassa, it's a filamentous fungus, and if you look at it under a microscope, you can see lots of little spores. So these are asexual spores formed by mitosis, and the wind can blow them long distances, so the fungus can spread rather easily. Now, what I want to do is point out that I want to try and color this here blue or dark blue. Let's see if it works. Yep. And I want to color these orange without causing everything to turn orange. So all these spores are orange. Okay. Now, in the early 1990s, researchers were trying to figure out, how can we get an organism to make huge amounts of a certain metabolite? And if you could do that, well, then you could use that organism to make lots of a chemical or pharmaceutical, and you could have it done rather cheaply. So this blue pigment right here, or purple pigment, let's say, in petunia was a good model to study how this could be done. Try to increase the amount of pigment in that petunia flower. These orange spores, this was another good model. So let's see if we can increase the amount of orange pigment in these spores of this fungus. And if we can figure out how to do that, well, then we can figure out how to make these organisms make lots of high amounts of any different metabolite. So we're going to start with these things we can see and learn how the process works, and then we'll be able to use this mechanism to make lots of whatever metabolite we're interested in. So one of the ways we could do this, or what they thought in the 1990s, was take the gene that encodes an enzyme that is needed to make the pigment and put in many copies of that gene. So let's say gene for pigment, and we're going to put many copies, or the scientists at the time were trying to put many copies of this gene back into the organism. So we'll have the native copy plus many transgenes, each with a copy of the gene in that organism. So often when we think of a transgene, we think of taking a gene from one organism and moving it to a different organism. Well, in this case, we're just taking the same gene in the organism and trying to increase the copy number by putting many copies of this transgene in the genome of the organism. In one case, it was petunia, the enzyme for purple pigment. In the other case, it was Neurospora crassa, the fungus, the enzyme for making the orange pigment. So sometimes it worked. Sometimes it worked. Other times, however, it did not work. So the opposite phenotype was found. So in the case of the purple petunia, it's not a very good petunia flower, is it? It didn't change the stem purple. But in some cases, it led to no pigment being produced. So we'd get a white petunia. Or scientists would, when working with the fungus, would get a fungus that made white spores. So this was kind of perplexing. So they take the enzyme involved in making the pigments and increase the number of copies of this enzyme in the genomes of this organism, or increase the number of copies of the gene encoding this enzyme in these organisms, and you get the opposite result of what they expected. So for several years, scientists didn't know what was going on here. This is so weird. There were some scientists called it co-suppression. There were lots of mechanisms that are models to explain what was possibly going on. But we now know that most of these phenomena where you saw the opposite of what you expected, in the case of petunia or this fungus example over here, or other examples where scientists were trying to do the same thing. What they were looking at, what they had discovered, was RNA interference. I guess what they were touching on was RNA interference, and it was scientists a few years later that actually figured out and got the credit for discovering the process. So before we go into the next level, or the next set of, before I explain the set of experiments that discovered RNA interference, precisely, or identified it exactly. So I want to talk about one more set of experiments that led to the hypothesis that RNA interference exists. So after those experiments there, and I should say around the same time, researchers were using a technique called antisense gene silencing. Now, essentially, how does this antisense gene silencing work? So if we have a gene, we'll call this gene A. Well, gene A is transcribed, let's say, into messenger RNA A. Now, messenger RNA A, we know by now, has a 5' cap, right? Has the coding sequence there, and then ends with a poly A tail. And I'll put some bases in here. So let's say this is the messenger RNA of gene A. Now, what antisense gene silencing is there were researchers who thought, what would happen if we took an antisense RNA? So we made an antisense RNA molecule that matched antisense AS RNA that was complementary to the messenger RNA of gene A. So it can hybridize to it. And okay, so if that could happen, would that silence the gene? So knock down expression levels of the gene by preventing it from being translated, right? Because that messenger RNA should be, ribosome should jump on there and translate it. But what if that antisense RNA is in the way? Perhaps that would block translation and prevent that protein from being made. So the gene would be silenced. We'd be knocking down levels of that protein by using this antisense RNA. And, this is 2020, and you might be like, why would we want to do that? We could just use CRISPR technology and mutate the gene. Well, way back then, CRISPR didn't exist. We didn't know about it. So it wasn't easy to mutate genes in lots of different organisms. And injecting organisms with an antisense RNA was easy. That was relatively easy to do. And, it turns out that this worked. This worked. And one thing I want to focus in on just briefly, just to make sure everyone is on the same page. If we were to zoom in at this little area right here, what would we see? Well, we'd just see, I'm going to make up a sequence here for the antisense RNA. So with a 3' end and a 5' end, and it would complement part of the messenger RNA. So it doesn't have to match the whole thing. So it just needs to complement part of it. I'm putting those dots there to indicate that the messenger RNA goes a little bit longer, and maybe the antisense RNA does too. And what they found was, sometimes this works. Sometimes it works. And that's kind of frustrating, the sometimes. So what they could do was inject these, or use some other technique to get these into cells, or inject them into an organism like a nematode or C. elegans, which I'll talk about in a moment. Inject antisense RNAs equals the silencing phenotype. Sometimes. And what do I mean by silencing phenotype? The phenotype that results when the gene product, whatever the gene is, when that product is knocked down. So now I can give you an example. Let's look at a specific example. So in C. elegans, which is a nematode, Caenorhabditis elegans, we can just call it C. elegans. So there's a gene called UNC22, and it is transcribed to make the UNC22 messenger RNA, which is translated to make the UNC22 protein. Now, in wild-type C. elegans, there's my little nematode here. This is a very small organism. You need a microscope to see it. So in a wild-type C. elegans that makes plenty of the UNC22 protein, well, it doesn't really do much. It just kind of moves around in the medium, or whatever environment it's in. But in UNC22 mutant, or a nematode that has had the UNC22 gene silenced, it will twitch. So the UNC22 gene was a useful gene to study gene expression, processes because it gave a nice phenotype to researchers to know when the UNC22 gene was silenced by whatever phenomenon. If the nematode was twitching, well, UNC22 protein wasn't there. So now with respect to the antisense RNA silencing, what researchers could do was make the antisense RNA for UNC22 in vitro, and there are in vitro techniques that researchers can use to make an antisense RNA molecule, and they can inject that into the nematode, using a fancy apparatus where you can inject these nematodes under a microscope. So, and we can get the twitching phenotype. So, however, you can get it sometimes. Some researchers could get it to work, others couldn't. Now, interestingly, it became kind of frustrating, right? When some labs are getting it to work and others aren't. Then others discovered that sense RNA could sometimes be injected into the nematode, and that would cause the twitching phenotype. So the model that was used to explain how antisense RNA based gene silencing was working involved hybridization of the antisense RNA to the messenger RNA and blocking translation. Well, that doesn't make sense, right? If sense RNAs are also working to cause silencing, well, that doesn't make sense because the sense RNA matches the messenger RNA. It doesn't complement it, so it can't complement to the messenger RNA. So we had a big problem at this point right here. So it took a couple scientists to figure out what was really going on. And they won the Nobel Prize, and I believe they were awarded the Nobel Prize in 1998 for their discovery of what was happening here. So their hypothesis was that the silencing phenotype is not caused by antisense RNA or sense RNA. It is caused by double-stranded RNA. Okay, so that was their hypothesis, and what they were also hypothesizing was that when a researcher uses the in vitro techniques to make, say, antisense RNA. So antisense RNA made in vitro, there is some double-stranded RNA contamination. So using the standard techniques of the day, when a researcher tried to make antisense RNA using in vitro techniques, there was often some double-stranded RNA contamination. And this was the trigger. This was the trigger of the gene silencing, not the antisense RNA. So same thing for sense RNA. When sense RNA was made in vitro, there was some double-stranded RNA contamination. And it wasn't the sense RNA that was causing the silencing phenotype. It was the double-stranded RNA. And it must have been especially frustrating because the more careful researchers who took extra precautions to make sure that their antisense RNA was pure, it didn't contain any contaminant, couldn't get the process to work. Couldn't get it to work. So it must have been very frustrating. It's almost like the sloppier the researcher was, the more likely they were to get antisense RNA or sense RNA silencing to work. And these researchers weren't really studying, the silencing mechanism. They were studying other processes that needed, and as their experiment, they needed to turn a gene off. They needed to silence it to study the gene. So it must have been a frustrating few years for researchers who were trying to use that technique to silence the genes they were interested in studying. So, okay, so what did Fire and Melo do? How did they, or Mello, I'm sorry, I still don't know how to really say his name. I'm going to say Mello, like Mellow Yellow, I guess. Fire and Mello. So, okay, so they studied the UNC22 gene, which is convenient because I already introduced the UNC22 phenotype to you. And what they did, they went through elaborate procedures to make sure that their antisense RNA was only antisense RNA, didn't have any double-stranded RNA contamination. So they used highly purified antisense RNA. And they used highly purified, you can probably guess what I'm going to say here, right? Sense RNA. And they used highly purified double-stranded RNA. For what? They just didn't use random RNAs here. All these RNAs matched the UNC22 gene. And so when they injected the pure RNA types into nematodes, well, those are my nematodes here. We got no twitching here, no twitching here, so no twitching ever here, never over here, and always with the double-stranded RNA. Oh. Okay, so yeah, so this experiment, they're awarded the Nobel Prize for figuring this out, because what did they discover? I think they actually coined the name RNA interference. RNA interference, which is a double-stranded RNA-based gene silencing process, and it's found in almost all eukaryotes, except for just a few. In one of the key eukaryotic organisms that does not have RNA interference is yeast. Well, at least the baker's yeast. The one that was the most commonly studied eukaryotic organism for figuring out how eukaryotic organisms work does not use RNA interference. So that's why, it went on for so long. If it had existed in baker's yeast, then I'm sure it probably would have been studied much earlier than and discovered much earlier than in the 1990s. So in the next video, we are going to take a look at the mechanism of RNA interference and how the various enzymes involved in the process. So one thing I want to touch on before I end this video is, okay, so we started with these other studies in the early 90s of gene silencing phenomena. So where scientists were trying to increase the amount of pigment in petunia, and they ended up making white petunia. And they tried to increase the amount of pigment in fungal spores, and they made white spores. So essentially what we think was going on is when they took the transgene, so the copies of the gene that encoded the enzyme that was needed to make the pigments, and they tried to put many copies of this gene in each organism. So the larger the numbers of copies in the organism, the more likely the researchers were to see these white phenotypes. And what we think is that, for example, let's say this is a random location in either petunia or the fungus Neurospora crassa. So this is a gene. This is just a normal gene in there. Here's the promoter over here. Here's the +1 transcription site, so the RNA polymerase goes that way, from right to left. Well, let's say the transgene inserted like that. So the transgene has its own promoter right here, and the transgene will be transcribed from left to right based on the presence of this promoter right here. But because it integrated right between this gene that was already in the organism and its promoter here, it will also be transcribed in the other direction. So at that locus, the transgene is going to be transcribed in both directions by RNA polymerase, giving rise to sense RNA and antisense RNA. So we get transcription of the sense RNA and transcription of the antisense RNA. And those can hybridize to make the double-stranded RNA. And then that can trigger RNA interference, which causes gene silencing of that native copy of the pigment-producing gene in each organism, Petunia or Neurospora crassa. So again, by trying to put many copies of the transgene in the organism, scientists would, or researchers would, increase the chance that they would get an integration like that could lead to production of both the sense RNA and the antisense RNA. And that's one model to explain what researchers were seeing back then in the early 90s. Other models have multiple transgenes integrating in a tandem array and sometimes flipping around. And then if they integrate many copies at the same location in opposite orientations, that can also lead to the formation of a double-stranded RNA that can then trigger RNA interference. Okay, well, in the next video, we will take a look at how the double-stranded RNA molecules trigger RNA interference and cause gene silencing.